Hereditary Hemochromatosis and the HFE mutation. On September 13, 2009, Malcolm Casadaban, a researcher at the University of Chicago, died at age 60 of flu-like symptoms. In his lab, he was studying the bacterial species Yersinia pestis, which causes the plague. While this organism was thought to be attenuated or weakened, it turns out his very own research subject is what killed him. Most people's immune system would probably be able to fight off this infection, but his was compromised. It was known he had diabetes, but upon further investigation and autopsy, it was determined he also had a condition called hereditary hemochromatosis. What is hereditary hemochromatosis? Hemochromatosis is a disease where too much iron can build up in the body. This is called iron overload. It is difficult for people with this condition to get rid of extra iron, and so it builds up in the organs, especially the liver, and can cause arthritis, organ damage, and cancer. Also, pathogenic or disease-causing microbes such as Yersinia pestis, shown here, can take advantage of this free iron for their own good and multiply, causing illness. So how does one acquire this condition? Most cases result because of a mutation for the HFE or the high iron Fe gene. The normal instructions for this gene prompt a complicated cascade of cellular reactions with the end result causing expression of a final protein called hepcidin. This protein allows dietary iron to be processed properly. In the diagram shown here, you can see the HFE protein in the membrane as it combines with the TFR2 protein and a host of others to elicit this cascade. Each person has 2 copies of the HFE gene, one from their mother and one from their father. People that have both copies of a mutated gene called C282Y, which I will explain later, are most likely to eventually develop this condition. It is an autosomal recessive trait more common in white males of Western European descent. The HFE gene, discovered in 1996, is found on chromosome 6, specifically on the short arm of the chromosome 6p22.2. Before we go on, let me explain a little bit about the Human Genome Project. Our entire set of DNA, 3 billion nucleotides, is called the genome. The Human Genome Project was proposed in 1988, began in 1990, and was completed in 2003, thanks to thousands of people in 6 different countries. Without this project, we would not know nearly as much as we do about many diseases, including hereditary hemochromatosis. You can see the goals of the original Human Genome Project here. Determine the order of all bases in human DNA, map the location of over 20,000 genes to their specific locations on individual human chromosomes, and also produce linkage maps, especially for disease that can be traced through generations. After the original goal was met in 2003, the National Human Genome Research Institute now conducts research to turn genomics into understanding human biology and developing methods for the detection, prevention, and treatment of heritable and genetic disorders. Many of the topics include these at the right. The Genome-Wide Association Study website has an interactive map of 24, including the X and Y chromosomes. The catalog publishes the GWAS diagram of single nucleotide polymorphism trait associations, or areas on the DNA that can be variable, mapped onto the human genome by chromosomal location as seen on the human karyotype. This is a little overwhelming to look at, but if you look at all of the dots on all of the chromosomes, basically all of these areas have been studied. While we know that DNA makes messenger RNA to assemble proteins, it doesn't hurt to remind ourselves with part of this clip, the Central Dogma song. This is one of my favorite videos to show students.
Started from induction, a simple path to follow. DNA to RNA, RNA to protein. The Central Dogma song. From DNA to RNA, RNA to protein. It's almost never wrong. Never wrong.
While we hear in the song that it's almost never wrong, sometimes it is. The normal HFE protein is made up of 383 amino acids or protein subunits. For the C282Y allele or mutation, at the 845th nucleotide, there is one tiny point mutation or substitution of adenine, an A, instead of guanine, a G. You can see over here that you have your amino acids tyrosine and cysteine. The differences are just your A and your G. Because of this one mistake in the DNA, the 282nd amino acid is tyrosine instead of cysteine. Proteins fold and bind in a very specific way. The HFE mutation causes this protein to be made incorrectly, and it doesn't work in the complicated cascade like it should. Diagnosis used to be obtained only from taking liver samples or measuring different levels of iron in the blood. While these methods are still used, there is an additional tool, and that is DNA testing. Because of the Human Genome Project, the HFE gene is known to be located on chromosome 6. Sequencing techniques have allowed for routine genetic screening, which can determine if someone carries this mutation. If you have heard of ancestry kits like 23andMe, anyone can find out if they have inherited the mutation. Women tend to develop symptoms later than men because of blood and iron loss through menstruation. Males that are homozygous for the C282Y mutation typically had health problems in their early 50s. The best treatment, but not cure, for hemochromatosis is phlebotomy or bloodletting. This should be started when serum ferritin levels are very high. Removing blood restores iron levels. At first, blood is removed on a weekly basis for a year or two, and then 2 to 4 times a year after that for maintenance. Serum ferritin levels are checked often. If a person cannot receive phlebotomy for various reasons, iron chelation, or removal of iron by drugs, is the next choice. People with this condition can follow a fairly normal diet, but should avoid a few things: iron supplements, excess vitamin C, which helps iron absorption, alcohol, which furthers liver damage, and shellfish, which tend to carry opportunistic bacteria that thrive on the excess iron. The incidence of the HFE mutation, homozygous for 282Y, is high, 5 in 1,000 people, but the expression is much lower. There must be other factors involved, including epigenetics and the environment. From an evolutionary point of view, most females of reproductive age are likely to be iron deficient due to menstruation and childbearing, so this mutation may have originally had some advantage to protect against this. Is genomics and the new technology available worthwhile to most people? That's up for individuals to decide. In the case of the University of Chicago researcher, DNA sequencing technology allowed for the cause of death to be determined. The specific strain of Y. pestis that was being studied was identified, and the man was found to be homozygous for the C282Y mutation that causes hemochromatosis. It turns out that this bacterial species, Y. pestis, can take advantage of its human host's extra iron. He was not aware of his condition because he had not been tested. If he had known, he could have easily been treated and his life possibly spared.